चैप्टर नंबर 40 डीएनए आइसोलेशन। आज हम लोग देखेंगे डीएनए को कैसे आइसोलेट किया जाता है। तो चलिए शुरू करते हैं। डीएनए एक्सट्रैक्शन इज द टेक्निक यूज्ड टू आइसोलेट डीएनए इन a biological sample by breaking the cell membrane and nuclear membrane with the help of chemicals, enzyme or physical disruption. The ability to extract DNA is of primary importance to studying the genetic cause of disease and for the development of diagnostics and drugs. It is also essential for carrying out forensic science, sequencing genomes, detecting bacteria and viruses in the environment, and for determining paternity. The first DNA extraction attempt was performed by Frederick Mischer in 1869. Later on, in 1958, Michelson and Stahl developed a full-function protocol for DNA extraction. The density gradient centrifugation protocol was the first protocol described by isolating DNA from Escherichia coli bacteria. The basic procedure for extraction is: first, cells which are to be studied need to be collected. Second, breaking the cell membrane open to expose the DNA along with the cytoplasm within cell lysis. Third, lipids from the cell membrane and the nucleus are broken down with detergents and surfactants. Fourth, breaking down proteins by adding a protease, optional. Fifth, breaking down RNA by adding an RNase, optional. Sixth, the solution is treated with a concentrated salt solution saline to make debris such as broken proteins, lipids, and RNA clump together. 7. Centrifugation of the solution, which separates the clumped cellular debris from the DNA. Okay, centrifugation 7 number. 8. DNA purification from detergents, proteins, salts, and reagents used during the cell lysis steps. The most commonly used procedure are ethanol precipitation, usually by ice called ethanol or isopropanol. Since DNA is insoluble in these alcohols, it will aggregate together, giving a pellet upon centrifugation. Precipitation of DNA is improved by increasing of ionic strength, usually by adding sodium acetate. Phenol chloroform extraction, in which phenol denatures proteins in the sample. After centrifugation of the sample, denatured proteins stay in the organic phase. While the aqueous phase containing nucleic acid is mixed with chloroform to remove phenol residue from the solution. Okay, phenol chloroform extraction, in which the phenol denatures proteins in the sample. After centrifugation of the sample, denatured protein stay in the organic phase. While the aqueous phase containing nucleic acid is mixed with chloroform to remove phenol residue from the solution. Mini-column purification, that relies on the fact that the nucleic acid may bind adsorptions to the solid phase silica or other, depending on the pH and the salt concentration of the buffer. Mini-column purification, that relies on the fact that the nucleic acids may bind adsorption to the solid phase silica or other, depending on the pH and the salt concentration of the buffer. 9 point, cellular and histone proteins bound to the DNA. Cellular and histone proteins bound to the DNA can be removed either by adding a protease or by having precipitated the proteins with sodium or ammonium acetate, or extracted them with a phenol chloroform mixture prior to the DNA precipitation, or extracted them with a phenol chloroform mixture. Phenol chloroform mixture prior to the DNA precipitation. Okay, 10th point is after isolation, the DNA is dissolved in a slightly alkaline buffer, usually in a TE buffer or in ultra pure water. This is cell membrane, nuclear envelope, and this is nucleic acid. Cell lysis of cell wall or cell membrane, lysis of nuclear membrane, nucleic acid comes out from nucleus. General outline of how DNA can be extracted from the cell. Different method of extraction. Some of the most common DNA extraction methods include organic extraction, silex extraction, and solid phase extraction. These methods consistently yield isolated DNA, but they differ in both the quality and the quantity of DNA yielded. When selecting a DNA extraction method, these are multiple factors to consider, including cost, time, safety, and risk of contamination. Okay, different method of extraction. Some of the most common DNA extraction method including organic extraction, silex extraction, and solid phase extraction. These methods consistently yield isolated DNA. These methods consistently yield isolated DNA, but they differ in both the quality and the quantity of DNA yielded. When selecting a DNA extraction method, there are multiple factors to consider, including cost, time, safety, and risk of contamination. Cost, time, safety, and risk of contamination. First, organic extraction. Organic extraction involves the addition of and incubation in multiple different chemical solutions, including a lysis step, a phenol chloroform extraction, and ethanol precipitation and washing steps. Organic extraction involves the addition of and incubation in multiple different chemical solutions, including a lysis step, a phenol chloroform extraction, and ethanol precipitation and washing steps. Okay. First, organic extraction involves the addition of and incubation in multiple different chemical solutions, including a lysis step, a phenol chloroform extraction, and ethanol precipitation and washing steps. Organic extraction is often used in laboratories because it is cheap and it yields large quantities of pure DNA. Though it is easy, there are many steps involved and it takes longer than other methods. There is an increased risk of contamination due to transferring the DNA between multiple tubes. Organic extraction. involves the addition of an incubation in multiple different chemical solutions, including a lysis step, a phenol chloroform extraction, and ethanol precipitation and washing steps. Organic extraction is often used in laboratories because it is cheap and it yields large quantities of pure DNA. Though it is easy, there are many steps involved and it takes longer than other methods. There is an increased risk of contamination due to transferring the DNA between multiple tubes. Okay, second is chillax extraction method. Involves adding the chillax resin to the sample, boiling the solution, then vortexing and centrifuging it. The cellular material bind to the chillax beads while the DNA is available in the supernatant. The chillax method is much faster and simpler than the organic extraction. The chillax method is much faster and simpler than organic extraction, and it only requires one tube, which decreases the risk of DNA contamination. And it only requires one tube. The chillax method is much faster and simpler than organic extraction, and it only requires one tube, which decreases the risk of DNA contamination. Which decreases the risk of DNA contamination. Unfortunately, chillax extraction does not yield as much quantity, and the DNA yield is single standard, which means it can only be used for PCR-based analysis and not for RFLP. Third, solid phase extraction, such as using a spin column based extraction method, takes advantage of the fact that DNA bind to silica. The sample containing DNA is added to a column containing a silica gel or silica beads and coatropic salt. The coatropic salts disrupt the hydrogen bonding between strands and facilitate binding of the DNA to silica by causing the nucleic acids to become hydrophobic. This exposes the phosphate residue or or this exposes the phosphate residues so they are available for adsorption. The DNA binds to the silica while the rest of the solution is washed out using ethanol to remove coatropic salt and other unnecessary constituent. The DNA can then be rehydrated with aqueous low salt solution, allowing for elution of the DNA from the beads. This method yields high quality, largely double stranded DNA, which can be used for This method yields high quality, largely double stranded DNA, which can be used for both PCR and RFLP analysis. This procedure can be automated and has a high throughput, although lower than the phenol chloroform method. This method yields high quality, largely double stranded DNA, which can be used for both PCR and RFLP analysis. This procedure can be automated and has a high throughput, although lower than the phenol-chloroform method. This is a one-step method, that is, the entire procedure is completed in one tube. This lowers the risk of contamination, making it very useful for forensic extraction of DNA. Multiple solid phase extraction commercial kits are manufactured and marketed by different companies. The only problem is that they are more expensive than organic extraction or chilex extraction. Once the DNA has been separated, it has to be tested for purity and also presence. Detection of DNA. Presence of DNA या diphenyl amine DPA indicator will confirm the presence of DNA. This procedure involves chemical hydrolysis of DNA when heated. Example, greater than 95 degrees Celsius in acid. The reaction requires a deoxyribose sugar and therefore is specific for DNA. Under these conditions, the 2-deoxyribose is converted to omega hydroxy levulinyl aldehyde, which react with the compound diphenylamine to produce a blue-colored compound. DNA concentration can be determined measuring the intensity of absorbance of the solution at the 600 nanometer with a spectrophotometer and comparing to a standard curve of non-DNA concentrations. DNA purity. Measuring the intensity of absorbance of the DNA solution at wavelengths 260 nanometer and 280 nanometer is used as a measure of DNA purity. DNA absorbs ultraviolet light at 260 and 280 nanometers, and aromatic proteins absorb ultraviolet light at 280 nanometers. A pure sample of DNA has a ratio of 1.8 at 260 to 280 and is relatively free from protein contamination. A DNA preparations that is contaminated with protein will have a 260 to 280 ratio lower than 1.8. DNA purity. Measuring the intensity of absorbance of DNA solution at wavelength 260 nanometer or 280 nanometer is used as a measure of DNA purity. DNA absorbs ultraviolet light at 260 and 280 nanometers, and aromatic proteins absorb ultraviolet light at 280 nanometers. A pure sample of DNA has a ratio of 1:8, 1.8 at 260 upon 280, and is relatively free from protein contamination. A DNA precipitation that is contaminated with protein will have a 160-180 ratio lower than 1.8. DNA can be quantified by cutting the DNA with a restriction enzyme, running it on a agarose gel, staining with ethidium bromide or a different stain, and comparing the intensity of DNA with a DNA marker of non-concentration. Using the Southern blot technique, this quantified DNA can be isolated and examined further using PCR and RFLP analysis. These procedures allow differentiation of the repeated sequences within the genome. It is this technique which forensic scientists use for comparison, identification, and analysis. Procedure: a spin tube method. In your fresh 1.5 ml centrifuge tube, adding 20 microliter of proteinase K. Second, transfer 200 microliter of whole human blood. Mix well by brief vortexing. Add 200 microliter of lysis buffer and immediately mix well by brief vortexing. Fourth, incubate at 56 degrees Celsius for 15 minutes. Centrifuge equilibrating. Adsorption. Centrifuge rinse. Elute. Centrifuge analysis. The image represents the general procedure of a spin column method for DNA extraction. Fifth point, add 200 microliter of 100% ethanol and mix well by brief vortexing. Fifth point, add 200 microliter of 100% ethanol and mix well by brief vortexing. After mixing, briefly centrifuge the 1.5 ml microcentrifuge tube to remove drops from the inside of the lid. Sixth point, pipette entire sample into a pure fast spin column. Centrifuge at 8000 rpm for 1 minute. Discard the flow and place the column back into the same collection tube. Okay, 7th point, adding 500 microliter of wash buffer 1 ethanol added to the pure fast spin column. Centrifuge at 800 rpm for 1 minute and discard the flow through. Place the column back into the same collection tube. 8 point, add 500 microliter of wash buffer 2 ethanol added to the pure fast spin column. Centrifuge at 12,000 rpm for 3 minutes and discard the flow through with collection tube. 9. Transfer the pure fast spin column into a fresh 1.5 ml microcentrifuge tube. 10th, add 100 microliter elution buffer to the center of the pure fast spin column membrane. Take care not to touch the membranes with the pipette tip. Incubate for 2 minutes at room temperature and centrifuge at 8000 rpm for 1 minute. 11th, discard the spin column and check the purity of DNA by using a spectrophotometer at wavelength of 260 nanometer. Discard the spin column and check the purity of DNA by using a spectrophotometer at wavelength of 260 nanometer. ओके फ्रेंड्स, ये चैप्टर कंप्लीट हो गया है। नेक्स्ट चैप्टर है 41, यूज ऑफ सैचुरेटेड, अनसैचुरेटेड एंड ट्रांस फैट इन फूड। इसे हम लोग नेक्स्ट वीडियो में कवर करेंगे। तब तक के लिए बाय। हंसते रहिए, मुस्कुराते रहिए और पढ़ाई करते रहिए। बाय बाय। थैंक यू।